Welcome everybody to week 4. This week we are going to be covering basically antibiotics or antimicrobials. Not the most exciting of topics. They can be quite complex, but we will be keeping this kind of as a broad overview level, not going too technical. We'll get there a little bit, but overall it's still going to try to just be a broad overview of the world of antimicrobials and how we test them and what we look for in the microbiology department. So we will start first with this PowerPoint. There's 2 PowerPoints. This one is called Antimicrobial Agent Mechanisms of Action and Resistance. So antimicrobial agents are just anything that inhibits or kills the infecting microorganism. It's in an active form. Again, we have to give sufficient concentrations at the site of infection to try to get rid of that microorganism. So depending on what level or amount you have to give will differ, depending on the organism and how pathogenic it is and how virulent it is, So different infections will require different concentration amounts. There also will be a direct interaction then between that antimicrobial agent and the microorganism to kill it, to inhibit it from continuing to cause infection. All right, so a couple terms that you do need to know right away is the difference between bacteriostatic and bactericidal. And the key part is going to be the suffix there, static versus cidal. So bacteriostatic is an agent that inhibits bacterial growth. It doesn't necessarily kill the organism, it just inhibits it from growing further and continuing to cause infection. Bactericidal, cidal means kill. That's agents that are going to actually kill the target organism. So those 2 terms I would note. So we categorize our antimicrobial agents on their mode of action. You see bolded there. So in other words, how do they go and inhibit or kill the organism? Do they target the cell wall? Do they target the DNA synthesis of that bacteria? Do they target folic acid pathways? Do they target the protein synthesis? Stuff that the bacteria needs in order to keep living and thriving. So there's always a mode of action, a target on how these antibiotics, antimicrobials, how they work. So they are trying to go in and inhibit or prevent a pathway, a synthesis, something inside that organism so that it stops doing what it was doing. So the most common pathways that are targeted are cell wall, cell membrane. Again, those are different things. They're not the same. Protein synthesis, DNA, and RNA synthesis. There are some that do folic acid pathways as well, which we'll discuss, but those are the more common structures that are targeted. So here's kind of a nice little graphic that shows it all together. So you have those that do cell wall synthesis inhibitors. So these are a bunch of examples. I know it looks probably small right here to you on this screen, but we can probably zoom in here a little bit. So you have like bacitracin, you have some glycopeptides. Vancomycin is a common example of a glycopeptide. That's probably one you might have heard of. Beta-lactams, that's like your penicillins, your monobactams, carbapenems, cephalosporins. Those are some huge antibiotics that have been prescribed over the years. So all of these are cell wall synthesis inhibitors. So they're going to target that outer cell wall area. Then there's the cell membrane synthesis disruptors. So lipopeptides, polymyxins like colistin are common cell membrane inhibitors. You have your folate synthesis inhibitors, sulfonamides, trimethoprim are the 2 that we're going to look at there. Protein synthesis, there is a bunch that fit into this category. Aminoglycosides is a big group. Gentamicin is especially a well-known antibiotic out of that group. Then you have a bunch of other groups down here. RNA synthesis inhibitors and RNA polymerase inhibitors, very similar. Rifampins, rifamycins, pretty interchangeable group there. They just, in the rifampin targets the RNA polymerase, whereas the others might do more just RNA synthesis in general. And then you have your DNA inhibitors, so fluoroquinolones, metronidazole there. So this is a broad overview, but it's nice to kind of see a little bit of a graphic interpretation of it. So what you're going to have to know here going in for the rest of this PowerPoint is the ones that we will discuss, you need to know their mode of action. What do they fit into, like for the category? So we'll start with those inhibitors of the cell wall synthesis. So again, glycopeptides are a big group in cell wall synthesis inhibitors. And you do need to know some examples that belong in the glycopeptide. Again, vancomycin was an example. The other one on here, I believe I gave was, teicoplanin. Not sure if I'm ever saying that one correctly, but it's right there, the second bullet point. But vancomycin is probably the most common example out of the glycopeptides. And so they inhibit the cell wall by blocking the formation of peptidoglycan. So we know that bacteria have peptidoglycan in their cell wall. So both gram-positive and gram-negative bacteria have peptidoglycan in the cell wall. So they will target the formation of that and inhibit it from forming peptidoglycan, so then you can form your cell wall. And so that will inhibit the organism altogether. So glycopeptides, though, only really target gram-positive bacteria because they are just too big. The glycopeptide itself is too big to fit through to get into the gram-negative more. So that is something essential to remember, is that glycopeptides are only targeting gram-positives. So, all right, the other big group out of the inhibitors of cell wall synthesis, again, is the beta-lactam. So that's your penicillin and all that. So they will target the beta-lactam ring. That's why they're called beta-lactam. So there is a beta-lactam ring that binds to enzymes that are used in the making of the cell wall. So they're targeting that ring, and so that way they can't make their cell wall. So again, a bunch of examples here to take note of: penicillin, cephalosporins, carbapenems, monobactams. So you see over here this kind of little image. So right here, this little square is highlighting that as a beta-lactam ring. So that ring is needed to help make their cell wall. So if they target that, they can, bust that open, and that way they can't form the cell wall. Now, because these are such a large group of antibiotics that have been used heavily through the years, a lot of bacteria have formed resistance to the beta-lactam drugs. So a lot of bacteria, penicillin doesn't work on them. Like, they're like, we've seen you, we now know how to fight you. So they have formed what they call beta-lactamases that prevent their beta-lactam ring from being targeted. So if they have the beta-lactamase ability, they can resist beta-lactam. So a lot of times these may not work for every organism because they've been so heavily used and they figured out how to block them. So that's kind of discussing that here. So resistance to beta-lactam. So again, they produce this enzyme called beta-lactamase. That enzyme will bind and hydrolyze the drug by opening up the beta-lactam ring. So they open up that beta-lactam ring in the drug, and it just doesn't allow it to bind to them and to take care of them. So there is a certain group of organisms called extended spectrum beta-lactamases, ESBLs. This is where it's getting a little bit more complex, but there's a bunch of gram-negative bacteria, especially, that are in the ESBL category, that they have a naturally occurring beta-lactamase enzyme inside of them. And so the new beta-lactam class of antibiotics that were developed to be resistant to these, but that bacteria, because they already had this naturally occurring enzyme, could still hydrolyze the drug anyway. So they came out with essentially a bunch of new beta-lactam antibiotics, like, hey, we got new ones that you haven't seen before that you shouldn't be resistant to. But these certain gram-negatives were still resistant to them because they still had a naturally occurring enzyme that was like, oh, we still can take care of you. So there's been over 150 of those ESBL organisms already identified that can do this. So that's pretty crazy that over 150 bacteria still are able to overcome those new antibiotics because they have that naturally occurring enzyme in them. So you have to treat them with something else. So again, just another little graphic up here of the beta-lactamase enzyme knocking open that beta-lactam ring, so that way it can't impede their cell wall synthesis and do anything to them. So that way they can resist the drug and keep thriving and causing infection. All right, now flipping over to cell membrane function. So again, cell membrane, very different than cell wall, keep them separate. A lot of people intermix them. So the cell membrane, the two examples here were the lipopeptides and polymyxins. An example of a lipopeptide would be daptomycin. They tend to target gram-positives. They insert themselves into the cytoplasmic membrane, just weaken it, leaking out the cytoplasm from the bacteria. So that will inhibit the bacteria. Polymyxins, a common example there is colistin. They tend to target gram-negatives. They can bind to that outer cell membrane that gram-negatives have. Remember, gram-negatives have an outside cell membrane that gram-positives don't. And so the colistin and polymyxins target that outside cell membrane layer. So that's why they target just the gram-negatives. So they are attaching onto that layer and inhibiting that cell membrane there, the lipopolysaccharide part. Protein synthesis. So they can inhibit protein synthesis by binding to ribosomes. Ribosomes are responsible for making proteins. That is something coming out of biology 101 when you first learned all your organelles. So ribosomes are responsible for making proteins. We need proteins to function, so do bacteria. And so by binding to the ribosomes, they can block and inhibit that protein synthesis production, therefore inhibiting the bacteria. There is, again, a ton that fit into this category. So you have your aminoglycosides, gentamicin, again, being a big example there, tobramycin, amikacin. You have your macrolides, which are some more mycins, erythromycin, azithromycin, ketolides, oxazolidinones, that's a hard word to say, chloramphenicol, tetracycline, and glycoglycines, all are here. As you can imagine, this is a lot of memorization. There's just, unless you can think of cute little sayings or figure out ways to remember all of these, which ones go into what mode of action, it's just a lot of memorization. Flashcards would be really helpful. When I was in school and I just made my flashcards by writing it out by hand, that helped me remember them, not even just practicing with them, but just making the flashcards always helped me. So on one side, you could put inhibit protein synthesis, and on the other, you could list these. Or you can list on one side aminoglycoside, and on the other, you have to name it inhibits protein synthesis. On another card, you can have chloramphenicol, and on the other, you have inhibits protein synthesis. So just make your flashcards, have somebody help you study them. There's just no easy way around memorization. So, you know, that's the problem with this unit, is it is a lot of memorizing. So knowing what each bacteria and examples, or each antibiotic and examples of antibiotics in those groups go with what mode of action. There was one cute little graphic I found on the protein synthesis inhibitors. I don't know if it really helps you remember them per se, but it's nice if you can go out and search for these kind of things. You know, there's a lot out there on Google, the interwebs, but whatever can help you figure them out. But so this was one graphic I found on protein synthesis inhibitors. Aminoglycosides, they have the gentamicin, tobramycin, amikacin here, and it says just call us the three amigos. So I don't know if the amigo part helps you with the aminoglycoside. I don't know how it helps you really remember protein synthesis inhibitor, but they have that cute little graphic. They have the tetracycline, things are about to get tetrifying, and they list a bunch of them that fit under there. But again, whatever graphics or sayings that you can come up with to help you memorize them. All right, continuing on, we have DNA RNA synthesis. So our DNA inhibitors are the fluoroquinolones and the metronidazole. Examples of fluoroquinolone, ciprofloxacin is a big example there, levofloxacin. And then rifampin is the one that would fit into the inhibiting RNA synthesis. So that one is easy for me because I just put the R's together, rifampin RNA. There really wasn't many other R names that we learned in this PowerPoint, so it kind of just always fit. And then again, the other metabolic processes, folic acid pathway can be inhibited. If you didn't take hematology where there was an anemia discussed that inhibited folic, if you didn't have enough folic acid, you weren't able to really make DNA. You need folic acid and vitamin B12 in order to make your nucleotides and DNA for your cells. So same thought process here. If we inhibit the folic acid pathway, we can't make our DNA ultimately, and the bacteria can't survive. So by targeting the folic acid pathway, they, in a roundabout way, target the DNA and then target this overall organism. So DNA directly targeted is by fluoroquinolones and metronidazole, whereas sulfonamides and trimethoprim inhibit the folic acid pathway. And then you have your nitrofurantoin that inhibits the citric acid cycle. This is a big one that is used to treat mainly UTIs. So nitrofurantoin is a drug that's mainly for UTI treatment. Okay, so that is a lot of knowing your mode of action, each group, where they fit, and the examples of each one. So mechanisms of antibiotic resistance. So again, requires interruption or disturbance of one or more steps is needed. So we have this biologic versus clinical resistance. Biologic resistant refers to that there's some change in the bacteria itself that makes it become less susceptible to that antibiotic, meaning the bacteria itself changed. You know, it had an evolution, it changed, so now it can resist the antibiotic. That antibiotic won't work on it anymore. Clinical resistance is when we have probably prescribed that medicine too many times, and now it's been out there and the effectiveness is lost, and they have formed a clinical resistance because we have just overdone it. And that is a very real thing that we are constantly fighting in healthcare to this day, is not right away jumping to antibiotics as prescription for anything. You know, it needs to be necessary. So many people just always want to go to the doctor and get an antibiotic, not understanding if you have a virus, that does nothing. But it used to be they just would give it to them because it was just something that they thought was going to help, or it would make the patient happy or whatever. But what it did in the long run is it create this clinical resistance that we're now finding that more and more bacteria are now resisting, you know, these medicines. And so you have to double up the medicine, like combo it with other medicines, go to bigger medicines that you don't want to always have to because of that. So that is clinical resistance. And then environmentally mediated resistance is a resistance that results from physical or chemical characteristics of the environment that that organism is that affects the organism and the drug and the response to the drug. So it could be different pH, anaerobic atmosphere, cation concentrations, things like that. For example, aminoglycosides are really affected by the concentration of calcium and magnesium in the environment. So there's other things that will play an effect on them. So this is more informational in this one right here. I do want you to know the difference between biologic and clinical. Environmentally mediated should kind of make sense by the name of it itself, but I don't think I have something directly off this slide. All right, and then you have microorganism mediated resistance. So this is the antimicrobial resistance due to genetically encoded traits of the microbe. So there is intrinsic and acquired. Intrinsic is a normal genetic physiological state that antibiotic never was able to target that pathogen. So something about that pathogen and that antibiotic just were never going to line up and work. It's just a natural state of resistance for however that bacteria is existing, that antibiotic isn't going to work against it. That's an intrinsic resistance. Acquired is it results from an alteration to the structure of that bacteria's genes. That's unpredictable. We never know when a bacteria is always going to acquire resistance and mutate and change. It just happens, and then we're like, okay, this has happened now. Now, you know, we have to figure it out. So intrinsic is just a natural, like they just were never going to work together to target it. Acquired is that bacteria changed and mutated, something changed in it, so now it's resisting it. So that's those two categories, knowing the difference between intrinsic versus acquired. Okay, so that's the first PowerPoint. Lots of memorization there. So going into the next one, antimicrobial susceptibility testing. So this is the testing we do in the micro lab. So besides identifying what is the organism that's causing the infection, you know, determining what bacteria we have, we also have to then report to the providers what antibiotics can be used to treat it. And that's where susceptibility testing comes in. We can give the provider a report of, okay, this bacteria will respond to these antibiotics. It is resistant to these. And that way it can help direct or guide their prescriptions and how they're going to treat that patient. So that is called antimicrobial susceptibility testing. You will hear the word susceptibility or sensitivity testing. They're interchangeable there. So AST for short. All right, so bacteria, again, can become resistant to antibiotics. And so we have to test for that resistance. So again, that's what we're calling sensitivity or susceptibility testing. So we're testing for that acquired resistance, that ability that they have to mutate and change their genetics and all of a sudden become resistant. So not every strain of bacteria and every infection is the same. They're all different. So every single time we identify an organism, we have to also send out a report of here's what you can use to treat it. So we do have a very standardized process, and that standardization is just to help control the environmental factors that are part of it. Because again, environment does alter or change bacteria. So we just said that with the cation concentration, the pH, that kind of thing. So we want to make sure that we're giving it the best growth conditions, the best ability to really give us consistent results, essentially, is the standardization process there, controlling those environmental factors. So there are very set guidelines that are out there published by the Clinical Laboratory Standards Institute, CLSI is short for Clinical Laboratory Standards Institute. Those guidelines are heavily followed by almost every micro department. They have committees of experts that get together and put together the guidelines that, show a lab, here's the ideal way that you should be doing your testing. Here's the ideal, things that you should be following. It helps, like it says, guidelines. It guides those micro departments. So there is an entire subcommittee on just antimicrobial susceptibility testing that every year, as they're learning new bacteria that become resistant to different drugs, they're updating those guidelines. They're updating what's considered resistant, what's considered susceptible, what level of concentration differs between them. There are levels and amounts that go with these things. So all of that is coming out by CLSI. They have an update every year on the antimicrobial susceptibility testing guidelines. So the labs, the micro departments, every year need to be updating their system and following these new guidelines that come out. So in the guidelines, they're going to have, again, the standardized process part. So how much inoculum are you using? So when you pick up your bacteria to do the susceptibility testing, we're looking for a certain inoculum amount. You can't just pick up however many you want that day. Like, oh, I'm going to take 2 colonies today. I'm going to take 6 colonies the next day on something else. Like, you have to have a certain concentrated inoculum amount. You should be using a specific growth medium. Typically, it's a Mueller-Hinton base. It's a very good supportive agar. That Mueller-Hinton we pretty much always just use for testing of antibiotics, disc testing, susceptibility testing. It's got a very well-known pH and cation concentration that just allows for good thriving on it. You should be testing the incubation temperature, the atmosphere, like if it needs CO2 or not. All of that is described in there. So, all right, now to directly measure the antimicrobial activity, so you need to bring that, of course, that bacteria and the drug together to see what the impact is. Will that drug work on that bacteria? So you are looking at either conventional susceptibility testing, which is usually using a broth dilution, an agar dilution, and discs, what we call disc diffusion. Nowadays, in our microlabs, we're using commercial susceptibility testing. So you're putting it in an analyzer. That's most microlabs are doing that, but there's still bench style ways of doing it like this. And maybe you might do that with certain organisms or certain reasons. There might be still rules in place of why some micro departments will still use disc diffusion for certain things. We will do some of this in our student lab because this is more bench side, whereas most things are getting run through an analyzer, and they're looking at a bunch of medicine at the same time, a bunch of different drugs at the same time on that bacteria and reporting it out. So, but there's 2 different ways there, the conventional versus the commercial now. So again, first you have to prepare your inoculum. For both ways, whatever conventional or commercial, you have to get an inoculum prepared. So you need to have a pure culture. So in other words, when you are picking out that bacteria, we're only looking at the bacteria we want to look at. If you have a bunch of other bacteria on that plate, you don't want to accidentally pick up a few different types of bacteria. Like if I want to look at this E. coli that I'm identifying on this plate, I know it's E. coli, I've identified it, and now I got its susceptibility tested. I don't want to accidentally grab a bunch of other stuff that might be present there too. And then you don't have a pure culture. You're now testing E. coli mixed with a few other bacteria. Those results are going to be so skewed on what comes out for what drugs work because you didn't have a pure sample. So that's what it means by pure culture is when you go to test it, you are only picking up the bacteria that you have identified and are knowing is the pathogen. So typically you're selecting 4 to 5 colonies of the same morphology and putting them into a broth. A lot of times we are doing what we call a 0.5 McFarland turbidity standard. So you are matching the inoculum size to this turbidity standard. So that just again ensures that you have like the right concentration of inoculum. So if we go to the next slide, it kind of shows you this. So there's a typo there. Used to compare, bacterial suspension to the McFarland standard. So before you put it onto a Mueller-Hinton agar, it's just a good quality control check. So a 0.5 is right here. So you should be able to see the lines behind it here. So they have a card that will come with this 0.5 McFarland standard. So you like take your 0.5 here, and then you take your sample that you made, and they should look really similar to this. You should be able to see the lines through here, through that cloudiness just enough after you made your inoculum. So your inoculum that you made should look similar to this level. If you get to where the lines are starting to become a little more obscure or just completely not there, that is too thick of a level. Now, there are times that you might use more than a 0.5. There might be times that a procedure calls for a 2.0 McFarland. Then you would compare it to that level. But most of the time, we are using a 0.5 McFarland. That is the ideal inoculum concentration. So it should look similar to this kind of level of turbidity. All right. So now we have a selection of antimicrobial agents to test. This is what we call an antimicrobial panel. So there will be different panels for different gram negatives and gram positives. So when we have a gram pos cocci that we've identified, we are testing it against known gram positive agents. You know, we just discussed how vancomycin glycopeptides are only good to target gram positives. So they're going to be on that panel. Whereas on a gram negative, we would never need to prescribe vancomycin for a gram negative organism. It's not going to work because vancomycin only targets gram positives. It's too big to get into the gram negative. So that's why there's a difference in antimicrobial panels. We'll have gram positive panels, we'll have gram neg panels. So again, it depends on what you identify is there. That will tell us the panel that we'll go to. Again, this is all given to us by CLSI. They have tables among tables that list potential micro agents, antimicrobial agents that should be in that panel. So they have ones for gram pos cocci. Here's the list that you should use for streps. Here's the list that you should use, you know, for Haemophilus. If you have a potential Haemophilus, test this panel. If you have Neisseria, test this panel. If you have gram negatives, rods in general, test these panels. So they have given us all that guidance on what should be included in the panel of drugs that you are testing to see, you know, if will work for that organism. So again, here's just an example. There are going to be testing panels or testing batteries for each of the following groups of organisms. And these, again, are all groups of organisms that you are eventually going to learn throughout microbiology, but we have different panels that we would use to treat different ones of these. So that is, again, given to us by CLSI. All right, so starting with a little description on some of these. Broth dilution. So you are using Mueller-Hinton broth instead of a solid or semi-solid agar. You are using a broth, so a liquid, but it's still Mueller-Hinton, so it's still a nice supportive broth to test. So you are going to make Mueller-Hinton dilutions in that broth. So typically it's a series of doubling dilutions, 16, 8, 4, 2, 1.5.25. So just like if you're taking clinical chemistry, you learned about serial dilutions. Maybe you've learned it in immunology as well. It's in there somewhere. I know we use dilutions in chemistry heavily. There's immunology also talks about dilutions, but you've learned how to at some point kind of discuss serial dilutions. That's essentially what we're doing here is we're making a serial dilution with our Mueller-Hinton broth and the organism of it. And you are looking for what we call the minimum inhibitory concentration, MIC. It is bolded for a reason. Know it. So what you are doing is seeing, okay, at what point does the drug stop being good to, you know, treat that organism? So at what concentration will the drug still inhibit that bacteria from growing? And whatever it is, the lowest amount of that is what we want to prescribe. We never want to give somebody too high of a concentration of too high of a dose of a medicine. We want to just give them the minimum dose needed to inhibit that bacteria. So that is what we're looking for is the minimum inhibitory concentration. What is the lowest level that will still inhibit that bacterial growth? That's the level that we are going to ideally use to prescribe to that patient. So you're essentially looking for the growth in each, you know, how did it respond in this dilution of the broth? So we can record it and translate it into 3 categories: susceptible, intermediate, resistant. These are what we kind of call like our breakpoint categories. All right, so up until this point, it's considered susceptible. At this point, it's right in between stages, intermediate. And then after this point, that bacteria is just resistant to that drug. It doesn't work at all anymore. So that's what it means by having those 3 categories. So obviously a provider is going to look at, okay, this drug is susceptible at this level. I can prescribe that level. So here's kind of a picture representation of this. So you have your dilutions being made. So like 2:1, you have 120 milliliters of broth, but you didn't put any antimicrobe in there. So no agent yet. It's just kind of broth only. Then we're going to put 100 mils of broth in there, but we're going to add 20 mils of antimicrobial agent. So the drug in there, not the bacteria, sorry. 0 of the drug there. 20 mils of the drug here, 40 mils of the drug here to 80 of the broth. So you get it. So you're making your dilutions. And then you incubate them because we have to let the bacteria grow if they're going to grow. So you have to put it in the incubator and come back the next day and look at it. So now the next day we take a look at it and we can see based on, for broth, you're looking again for turbidity. Turbidity, cloudiness always tells us the bacteria grew. So if it's got a ton of microbial growth, it'll be really turbid, meaning that bacteria thrived. So in the first tube, when there was no drug in there, of course your bacteria is going to be highly present. It was just the bacteria with the broth. In tube 1, there was no drug, no antimicrobial agent yet. So of course that's going to be super cloudy. In tube 2, now you can see, okay, we added some antimicrobial agent, but there's still some cloudiness there. It still wasn't enough to treat and inhibit that bacteria. That bacteria is still thriving and so on. So in this case, on these 7 tubes, for example, the very first time that the bacteria was finally inhibited was in tube 6. There was no cloudiness, no change. It totally inhibited that bacterial growth. Same with tube 7 also works. So those levels of concentration worked on the bacteria. Now, which one is the MIC? The minimum is going to be tube 6. That is the first level that it finally inhibited at. That is the lower concentration. We are going to prescribe that level. You know, yes, 7 worked as well, but why would we need to prescribe level 7 if level 6 worked? It's a little bit less amount. So like up here, we had 100 milliliters versus 120. So 100 milliliters is going to work just fine for that patient to prescribe. So that is the minimum inhibitory concentration level there. All right, you can also do basically the same thing, only on agar. So that was broth dilution. You can also do it on agar plate. So on a Mueller-Hinton agar. So again, in that agar, you have antimicrobial concentration and organisms brought together on that agar. You're still using dilution, doubling dilutions. You're still inoculating the plates and then incubating them overnight. And again, examining them to see what has the lowest concentration that prevented the bacteria from growing. So it's the same setup, basically just on an agar plate instead of in a broth. And then you have the disc diffusion. So this is what you will probably play with a little bit in our student lab for sure. This is commonly known as the Kirby-Bauer test. So disc diffusion, Kirby-Bauer, same thing here. So you are using little antibiotic discs that you place on the Mueller-Hinton agar. So you have your Mueller-Hinton agar. You're going to have little antibiotic discs. So whatever disc that you want to test, if you have like say a cephalosporin disc, a erythromycin disc, you know, a tetracycline disc, you know, you can have all sorts of different antibiotic discs that we want to put onto the plate. We're streaking basically a lawn of growth across that plate. So a lawn of growth meaning the whole thing is really covered there with, you know, we make our inoculum still, we figure out the bacteria, you know, make your inoculum standard. And then you're using that inoculum standard and streaking out the plate and you have your antibiotic discs around it. So as it says here, you place those discs on the agar plate with a lawn of bacteria. So after you make your inoculum, you streak out your lawn, place the discs on there and all that kind of thing. And then you incubate it again overnight. The next day you look at it and you're looking to see, is there a zone of inhibition around each disc or not? So a zone of inhibition would look like this. So after you look at it the next day, the bacteria grew up until right there, and then that entire area, what's white around that disc, is showing a zone. That means that that antibiotic was able to kill off the bacteria and prevent them from growing in that area. So it worked. So you measure it in milliliters, and each antibiotic will depend on the measurement, on if it's considered susceptible, intermediate, or resistant. There is different millimeter amounts that go with each antibiotic disc, but you measure it and you might call this one, oh, this one is considered susceptible. So that bacteria is susceptible to that antibiotic still because it had this huge growth of inhibition here. There was no way that it could get up even close to that disc. So that drug is working really well on that bacteria. This one has a zone, but it's a smaller zone. So it just depends on the size of it. This one, there's no zone. That is what we call resistant. The bacteria didn't care about that antibiotic at all. That they were resistant to the antibiotic, so they grew right up to the disc. It didn't affect them whatsoever. So that would be what we call resistant. So if you look at a little bit more on this Kirby-Bauer test, again, you're using a Mueller-Hinton auger, you're making your inoculum, comparing it to the 0.5 McFarland standard to make sure you have a standardized inoculum. So again, you're going to make it look like this with how the lines are visible behind your, a certain level of turbidity. So you can try to mimic that as best as you can to have a standardized inoculum. Then again, like I said, you streak a bacterial lawn across the plate. So to streak it, you're kind of rotating the plate 3 times. You're, dipping into that inoculum and you streak heavily back and forth across the entire plate, rotate the plate a little bit, do it again across and rotate it a 3rd time and do it a 3rd time across. So that's making the bacterial lawn. Then you place all your discs on it. So in this case, there is a neat little antibiotic disc dispenser here that they have loaded so that in our lab, we might use like tweezers. You might sterilize your tweezers, let them cool, and then take a disc out and then gently place it on the plate, kind of tap it on, 1 each time. But you can see how long that could take because there's how many of these discs here. That takes a little while to do. Whereas in a lab, they might have a disc dispenser like this that has them all loaded in there, and then they just kind of push them down into it and it does it. And then they just kind of tap them into place. But again, you're going to incubate and then look for zones of inhibition. So you can see there's quite a few zones here, and you have to measure each one of those to determine, okay, is it going to be susceptible, intermediate, or resistant? Like this one here has a little one, but that might be called resistant because it's not big enough. It might say, you know what, that wasn't enough of a zone yet. We're still calling that resistant. Even though there's a little one there, it wasn't enough of a zone. Whereas they might definitely call some of these more susceptible. Maybe this one's intermediate here. They're like, it's not quite susceptible, but it's not resistant. It's in between, it's intermediate. So if we don't have to use that drug, let's not, kind of thing. So again, you measure each one of these and then you compare it to the tables that CLSI has given us to determine that. And that's kind of what we consider those breakpoints. And then, like I said, there's commercial testing where the instrumentation does it all for you, which is a beautiful thing. You have these cards, like this is one type down here. These are all different antibiotics. They can have inoculum here feeding into the card, and then they put it in their instrument, and then they look to see if which antibiotics, it reacted to, didn't react to, kind of thing. And they report all that out for you. You get a report from the instrument that does that. So Vitek, Microscan, Phoenix systems were all very popular. Sometimes it might require overnight. Sometimes it might be a few hours. It just depends on the analyzer. But again, they are comparing it. You program the computer in that system on what is considered susceptible, intermediate, resistance for each antibiotic in each, organism. So in this case, identification card types for that one, this is for the Vitek especially, I pulled this from, so Vitek instrument. They can identify and test 170 gram-negative bacilli, 128 gram-positive cocci and gram-positive rods, 117 anaerobes, 36 different Haemophilus and Neisseria, which that's huge because those categories aren't big, 60 different yeasts. So out of their gram-negative rod stays, so they can identify 170, they can have 85 antimicrobials tested, like on those cards. That is a lot of different drugs that we can test for. So you can see that's much more sensitive, much more accurate for the patient care than our little disc. Like we're not going to read 85 discs on a Kirby-Bauer. So again, there's still reasons that we use Kirby-Bauer in the real world. They definitely still have a place, but most facilities also have this in place because there's just so much more complexity to drug prescriptions and resistance now. All right, so again, AST results interpretation. So AST here again stands for antimicrobial susceptibility testing. It is not your liver enzyme that you might have learned in clinical chemistry. It is antimicrobial susceptibility testing in the world of micro. So again, I have been saying the word breakpoints. They describe the breakpoints. That is meaning what is the concentration that considers it still susceptible? What's the concentration now that's considered intermediate? And what's the concentration that's considered resistant? Every drug has a breakpoint specification. So penicillin for certain, bacteria are going to have susceptible, intermediate, resistant concentrations that are different than penicillin for a different bacteria. Same with like cephalosporin is going to have different categories. All the different drugs will have different levels of breakpoints. So those again are just specific concentrations that are describing it as either susceptible, intermediate, or resistant. And so again, those panels have all of that programmed into. And so your analyzer, your commercial analyzer might give you a result that looks like this. So they're going to report it out. So in this case, when I pulled this, so this was an Elizabeth Kingia meningoseptica. Oh my gosh, that's a complicated organism name. Not a familiar one, not a very common one, by the way. That is not very common. So that was the organism that was causing the infection that was identified. And then down here, you have, here's your susceptibility information. So what's going to the provider is this. So they can see, okay, this drug or this bacteria is resistant to ampicillin, resistant to amoxicillin, clavulanic acid. It's resistant to piperacillin, cefoxiamine, ceftriaxone, you know, it goes on. And you can see the MIC, the minimum inhibitory concentration. Anything greater than 32 is considered the breakpoint of resistance for ampicillin. Anything greater than 128 was considered the resistance for piperacillin tazobactam. Anything greater than 16 was resistant for imipenem, that breakpoint. So they do have different breakpoints that CLSI has all determined. And so these are still resistant. So when I go here, which one am I going to prescribe for this Elizabeth Kingia? I guess I'm down to trimethoprim sulfamethoxazole. Everything else does not look very usable, right? So to treat this patient, I can use a minimum concentration level of 40, and the bacteria should respond to it and should be killed off or should be inhibited from growing, and that should hopefully clear up their infection. So that is how you're kind of putting it all together. These, you know, this information that's being fed back to you, you know, we are reporting this to the physician so that they can, again, if they had the patient on something else in the meantime, because this takes time. This isn't the same day always. You know, this takes time to get this info. So that doctor has probably prescribed something in the meantime. But now when this report comes back, whatever they prescribe, say they prescribe ciprofloxacin, well, now they're like, well, shoot, it's resistant to it. That's not working. They need to switch that drug that the patient is on to something that is going to work. Okay, and then antimicrobial combinations. You do have antimicrobials that are synergy versus antagonism. Synergy is that drug works better together with something else. So the drug on its own works okay, but it works really well with this other drug. It's synergistic. The combo of the two work really well together. And you'll see that there's a few here that are comboed up, just meaning they thrive together. They do better together than they do on their own. So that's synergy. Antagonism is a drug that's like, I am more effective on my own. If you combine me with something, it's not going to work as well. I don't like it. I want to be on my own. It's kind of like extrovert, introvert kind of thing. So that's what those kind of terms mean when they're describing antimicrobials. Okay, so that is the land of susceptibility testing. It can be quite complex to understand. Playing around in the lab with some of those discs and interpreting the zone of inhibition, that will help you kind of put it to the test. It is also, unfortunately, a lot of memorization, knowing some of these key terms that we defined. Do your best, make flashcards, find cute little graphics out there or sayings, whatever it takes to stick this information in your head. All right, thanks so much and have a great rest of your day.